Chetapalli, who, when he's not an emergency room physician or researcher or writing, he's actually also CEO of Sirica Therapeutics, which he's going to tell us about today. And it's a really interesting concept here that's probably unlike anything we've seen so far. So I'll leave it to Dr. Chetapalli to tell us about what he's doing. Thank you.
Thank you, Stuart. Can I share my screen? Great. Okay. Let's see, let's go back to the top. All right. Thank you very much. Thank you to the Brain Foundation, and thank you, Pramila, for giving me an opportunity to present Sirica Therapeutics. And I wanted to let your audience know that this is the first time we are actually introducing this concept and this company. So this is the launch of Sirica Therapeutics in a way. Now, I was looking at the date and I was thinking 12122021. Is that a code for something, some patent? We'll find out. But it's a great date because today I'm going to talk about Sirica Therapeutics. Let me put it on. Okay. I wanted to tell you about our daughter. Her name is Siri. She has autism. She was diagnosed when she was 3 years old. And like everybody else, we had to struggle to find what exactly needs to be done. One of the biggest things out there at that time was early, but it was ABA therapy. And, we followed the doctor's instructions and it did help. Although it was very painful, 20 to 30 hours is a lot of struggle. But we know that she has made some progress with that therapy. But later on, when she was done with the therapy, she was done with school, although she had a very difficult time during schooling, including difficult behaviors, aggressiveness, and injurious behaviors. So right after she graduated from school, when she turned 22, we did not have anything else to fall back on. The day programs were really bad and it did not fit her picture and it did not fit her needs. So one of the things we started doing was, working with her ourselves, my wife and I. What we noticed is that when we engage her, when we encourage her, when we challenge her, and when it is a gratifying solution for her, she works great. In fact, she makes big progress. She has now a business of her own running designsbysiri.com. She makes jewelry, she makes crafts, she does all these things. And people ask us, how did you do this? How did that happen? And so when I started back thinking about this, these are the experiences or the characteristics of these experiences that we have used that helped her improve her behavior, her mood, and her motivation. So I thought, can we put this into a device? And that search has now culminated in Sirica Therapeutics. The purpose is to develop and commercialize a technologically advanced, clinically proven, scalable therapeutic system for autism that is fun for the user. The fun is an important factor here. By the way, we are in Silicon Valley, and why not? The problem is the current treatments involve manual one-on-one interventions. It takes a long time, 20 to 30 to 40 hours a week. That's a long time. And the treatment is variable depending, it is operator dependent or therapist dependent. And most importantly, it is not fun because, we'd have to bribe the kids, or, we have to bring them back when they're screaming and complaining. So what are the components of this kind of a system? So we put together 3 components that work together to form this system. First one is virtual reality. And you have seen other places where they've used this. It's creating that deeply immersive environments that are stimulating and challenging. And then we added advanced robotics. Now, the robotics part is not just a robot that comes and talks with you. It's the whole device is a robot that you sit on and operate. So it has sensors and it can control the movements. It can add and add more inertia to the machine and to different parts of the machine. The third piece is where we collect data, thousands of data points from each session. And then imagine having multiple machines and we are collecting this data and using that data, applying machine learning algorithms to make the therapy more effective and the outcomes better. I wanted to show you the video. I can describe the machine, but it's better to see it yourself. This is Siri on the machine. What she is doing is she's writing this, as you can see. She is seeing this in her virtual reality goggles. And so she's technically riding a vehicle. There she hit a rock. So she's trying to get out. And this is the first time she's writing it. She has never written this before, and she's able to manipulate it. And after it was done, when I asked her, do you want to write some more? She said, yes. So this is the second session that she's writing on. As you can see, the machine itself is moving. So she can feel the bumps in the road and the rocks that she is hitting. So that is an interesting experience because we were developing this actually for stroke. But we had an idea that this machine could potentially be used for autism and many other similar neurological conditions where you need to put people in an environment where there is urgency and also it is a task-oriented therapy. And the idea behind this is that this will stimulate neuroplasticity. And I don't want to talk more into the science of it, but I will have that at another talk. So what we have done is it took a few years for us to develop this prototype. It took about 4 years to actually fine-tune this system so that the software, the hardware, the robotics, all these pieces work together and can give us enough data. So what's happening right now is that we are taking this as a separate company. Sirica is going to be purely focused on autism. Torque 3 is the original company, and that will continue. And I've been their chief medical officer for the last 2 years. Now, you must be thinking, where is the competition here? Well, I kind of did a Google search trying to find competitors in the robotic space, the virtual reality space, the machine learning space, and there has been some progress. For example, Yale University researchers are trying to do job training using a robot, but this is a robot that talks to you and is more like a teacher. I've looked at Florio, which is a VR learning system. And this is where you put on your VR goggles and it walks you through several lessons. And same thing here, adaptive technology, They have a driving simulator where you put on your VR goggles and you can drive. But it is not the same as what we are trying to do, where we are trying to combine all three. Cognoa, some of you may know, they have a digital diagnosis and treatment system using videos and iPhone. Achille is very interesting because they have used a video game to treat ADHD, and it was approved by the FDA. So this is just a video game that you hold and play. Now, what if you combine all these three different mechanisms and put it together in one piece? And that's what Sirica Therapeutics is trying to do. Now, we all know the economics, right? You know, there have been some great speakers, and there is this urgency that, you know, autism is growing. It used to be 1 in 54, and now it is 1 in 44 births, just 2 years ago. FDA has a very favorable regulation process now for this disease because of the urgency. And right now we have coverage in 50 states for therapy. Now, the main therapy is the behavior and the speech and OT, where we will need 60,000 therapists to be able to manage the demand right now. And the demand keeps growing. But right now we have only 30,000 therapists that are working. So we need 30 more. So there is such a high demand. And of course, the market size is going to grow. By 2025, it'll be 461 billion. So I put together a team, myself. My name is Yuli Cheripalli. I am a physician by training. I practiced emergency medicine at Kaiser. I have a background in epidemiology, population health, a master's from UCLA. Most of my career, I was at Kaiser. I developed systems, technologies, and platforms, which are now working in 21 hospitals, emergency departments. I worked at Anthem AI, designing AI systems. I worked at Torque3, being their chief medical officer. And also I created a community of physician innovators at innovatormd.com. Now I am taking on this bull by the horns because this is very personal to me because of my daughter. I have Kristen Jacobson, an amazing person. She and a few other parents got together several years ago and changed the laws. They went to the Senate in Sacramento and wrote these laws that hopefully one day will change the coverage for autism. And they were successful after 4 years of working diligently on it. She also worked at Autism Speaks, McKinsey, and she has an MBA from Stanford. Hiren Patel, you know, he's a biomedical engineer. He has worked from Rutgers. He's worked at J&J, Zimmer, Biomed, and Cook Medical. And Tim Lacey, he's our science guru. He has experience at Walter Reed. He's a psychiatrist by training and also a US Air Force veteran from GW University, George Washington. And he's the chief science officer at Torque3. He is helping us design the system for autism. And one thing I didn't tell you is that 3 of these team members have kids with autism. That is myself, Kristen, and Tim. I can tell you about projections of financial projections, but it would be very unrealistic. So I'll let somebody else talk about it. Some of you in Silicon Valley may recognize this person, but I'll play the video for you. Oh, it skipped.
Enormously lucky Sid and I have been as parents. The kids have been through, a lot. But we had the ability, we had the means, and we had the will and the luck to go out and find the AP and folks who are just technically and in every way just the best in the business. And there aren't that many people in the world who have access to this kind of care, or maybe are even aware of it. And when you look at, the best in the business, autism is turning out to be big business. So this is where I go from the parent version of the talk to the business version of the talk. So if you look at this stock, I ran a publicly traded company, Expedia, for 16 years. This is and we're rumored to go public, the new company that I'm running. This is a chart of the stock market over 40 years. And the blue chart is the Dow Jones Index. The red chart is Coca-Cola, one of the best performing stocks in the market. And then there's a new entrant, which is autism diagnoses, beats them all, right? So if you look at autism diagnoses as a growth industry, this is the ultimate growth industry. It's extraordinary how fast the rate of diagnoses has increased over the past 40 years. And Ron and I were talking about it. Is it that the definition, the spectrum definition, has expanded? Is it now that there's just much more awareness of the spectrum? So there's more awareness, there's more testing, et cetera. It's probably a combination of all those, right? But the fact is that this business, if you want to look at it, has gone from being a small cottage industry to being big business to being industrialized to some extent. We're going from a cottage industry to the Ford Model T that has been mass produced to the Prius.
Okay, so this is the ask. What we are doing is we are raising our seed round, a $2 million seed round. And we're looking for clinical partners to host and run our pilots. We will have 3 machines later in next year, and we need to run some testing and pilots, and we need research partners to be able to design and conduct studies. Thank you very much. I will stop there, and if there are any questions, I'll take it.
Sure, thank you, Yuli. So we have one question here, and then we'll have to move on. The question is, one of the challenges with any virtual reality application for autism is that a reasonably large subset in autism has sensory sensitivity and cannot wear the standard headset for a meaningful period of time. Have you explored if any of the headsets that may be easier to use for this segment?
Not yet, but that is a challenge, definitely. So we have to figure out how do you ease them into a headset. First, maybe we do have large screen areas where the child or adult is looking at the screen while they're riding this machine. You could do that. And as they get interested, as they get engaged, slowly introduce them to the headset. Now, the other factor is that you can control the full environment inside the game. So you can make it as blank or as less distracting as possible, or as less disturbing as possible. Is it the sound? Is it the colors? Is it the way it is designed? So we can slowly introduce them into an environment which is therapeutic. So that's what we are planning to do.
Great, thank you. And there are a couple of other questions in the Q&A that in the interest of time, we should just move on, and maybe you could answer those questions directly in the Q&A. That would be great. Sure,
thank you very much.
Sure.
Yes, let me take over here and introduce Dr. Campbell. He's CEO of Axial Biotherapeutics. Stuart Campbell is Senior Vice President of R&D at Axial Therapeutics and Japanese. He has consulted for several startup companies in multiple capacities and is a co-inventor on more than 15 issued patents. Thank you so much for consenting to chair the session. So take it away, Stuart.
Thank you, Pramila, and thank you again for the opportunity to present here again. And I hope you can see. You should.
Yes, I'm seeing the 2-slide version.
Okay, let me go to. Hang on, let me stop share for a moment and reshare in a different way. There we are. Is that better? Okay. All right. Well, today, earlier today, we kicked off with a talk from Sarkis Mazmanian from Caltech. And he talked a lot about the mechanisms that may be at work in between the microbiome and the brain with respect to metabolites that are produced by bacteria in the gut. And he mentioned that we would tell the next chapter of the story, which is we're beginning to translate those concepts into therapies. And I'll tell you about where we are, how we're approaching it, and where we are in this area. So quite excited to tell you. Why can't the slides advance? There we go. We'll tell you a lot about what we're trying to do here, in particular in the area of irritability associated with autism. And so as a company, we have more programs than the one I'm about to tell you about, but we're really trying to change the way that people think about treating neurological conditions, and we have some effort in cancer as well, by exploiting the way the gut microbiome interacts with us, the human host. And so just from a high-level perspective, Axial is a clinical stage company, and we're spun out of Caltech, founded by Professor Sarkis Mazmanian, and our job is to take his basic foundational science and bring it into the clinic in hopes of developing safe, effective therapeutics for various indications, including some of the co-occurring conditions that people with autism experience. And so what may be different from other companies that focus on the microbiome here is that we actually use the microbiome as a way to find new targets that could be amenable to therapeutic intervention. And so our interventions are actually gut-targeted small molecules that have very little to no systemic exposure. So these things are delivered to the gut, they transit through the gut and work on targets in the gut and affect things downstream like behavior, and we're starting to understand how that could be possible. So if we think about the different avenues of communication between the gut and the brain, we think about the microbiome gut-brain axis, and there are really three major highways here that we could group various mechanisms together. One is, starting from the left here, is the way that the gut microbiome can interact directly with the enteric nervous system by producing certain factors, protein aggregates, and so on. And this is actually the approach that we're taking to interventions in Parkinson's disease and other neurodegenerative diseases. In the middle, where we'll focus today is really on microbial metabolites that are produced exclusively by microbes in the gut that enter circulation and ultimately reach the brain and can alter brain cell populations related to certain behaviors. And lastly, the one that we've heard an awful lot about today is really the interaction between the gut microbiome and the immune system. So the gut microbiome educates our immune system on a lifelong basis, and that can have profound impacts on things like neuroinflammation and other various processes that result in different types of conditions that need to be managed. And so to focus here on autism, though, which again is really the central avenue here that we're exploring in autism, is the communication between the gut and the brain through metabolites that are produced by gut microbes that go through the blood and get to the brain. I won't go through this. We've heard this multiple times today about some of the background in autism, but I think what we should focus on here is that on the right-hand side, there are several behavioral interventions that are very helpful for certain segments of the autistic population. However, some require more help and support. And so there are painfully few options, drug pharmaceutical interventions that are safe and effective in this population. So there's large unmet needs in this population. And in fact, the only two approved drugs for anything to do with autism in the US are two atypical antipsychotics that are prescribed to manage irritability associated with autism. Given the safety profiles of these drugs, we think this is still a very significant unmet need. And so this was an area that we wanted to explore further for a variety of reasons that I'll show you as we go through this. So our motivation and rationale here is really, again, that there's very little from an option standpoint for parents and families. We know that GI disturbances, and we've heard a lot about this again today from several presenters, that GI disturbances are very prevalent in this population. The gut microbiome in children with autism is different compared to control populations. And these differences, and we'll show a little bit of this, can result in alterations of the metabolite profile that we see in certain segments of the autistic population. And so what we've tried to understand here is whether these metabolites could actually be contributing somehow to various manifestations of autism or its co-occurring conditions. And then therefore, could that offer new avenues for intervention? And so our mechanistic hypothesis here, and we heard a lot of the science behind this from Sarkis Mazmanian's talk earlier today, is that there's a class of neuroactive microbial metabolites. Sarkis discussed in depth the profile of 4-ethylphenyl sulfate, or 4-EPS in this regard, that they can contribute to autism-related behaviors by altering myelin plasticity in the brain. So that work so far has been in mice, but there is also an emerging set of literature that now implicates myelin plasticity in all sorts of behavioral changes and responses in various populations. And so we think that there's maybe a link between these two fields that warrants further investigation. Our treatment hypothesis here, if these metabolites are actually pharmacologically active, then we may want to try to reduce those as a way to impart a therapeutic effect. And so our approach really is to reduce these neuroactive microbial metabolites in the gut where they're produced, to reduce their systemic levels, hence their brain levels, which in turn should alter or improve problematic autism-associated behaviors. And so this goes back to tell you a little bit of the history, and we saw a little bit of this morning, is that our first clue here was the seminal publication by Sarkis and his group in 2013, wherein using the maternal immune activation model in mice, they identified in an untargeted metabolomic screen elevations or alterations, I should say, in several metabolites, some elevated and some depleted. But the most dysregulated metabolite in this case was 4-EPS, and it was significantly elevated in the mice that had their mothers mock infected with an immunostimulant during pregnancy. And what this seemed to correlate with were certain behavioral features. So what they did was it took it a step further and said, if this, perhaps this could be a biomarker, but let's see if it's pharmacologically active and maybe contributing somehow to the behaviors we're seeing in these mice. So they took synthetic 4-EPS and injected it daily into wild-type mice, and they were able to induce a pretty significant anxiety phenotype. Now, people in general don't get injected with 4-EPS on a daily basis, so this was a good proof of concept, but did not offer a more natural way of being exposed to this type of metabolite, which would be on a more chronic basis through the gut. But first, we wanted to take this and try to understand whether there was any relevance to the human population. And so earlier this year in biological psychiatry, we've had Paul Ashwood and Sarkis both talked a little bit about this earlier today. We did see elevations of 4-ethylphenyl sulfate in a significant number of the autism population compared to controls in the UC Davis Charge sample database. This was work that I said we published earlier this year. And so what we showed in this was that 4-EPS levels were shown to correlate with certain socialization deficits and different scores, and therefore it lent some credence to this argument that these metabolites may be biologically active. And so our overall approach here is that, or our hypothesis, I should say, is broader than 4-EPS, that there's in fact a class of structurally related metabolites that we call, get the pointer here if I can. Let's see. That such as these that are produced by microbes in the gut. They are rapidly absorbed into circulation. They go to the liver. The liver tries to get rid of them by modifying them chemically into things like indoxyl sulfate, paracresyl sulfate, and 4-EPS. And it turns out that these are the implicated molecules that we believe are contributing to certain behavioral changes at least in mice. And so the question is, does that translate to humans? And Sarkis talked about his model this morning, wherein they had developed mice with engineered microbiota that either produced or didn't produce the precursor for 4-ethylphenol, and then they could measure the differences in behaviors and several other aspects of brain function, brain transcripts, cell populations, and so on. And that's where the hypothesis that something like 4-EPS can arrest oligodendrocyte maturation, leading to alterations in myelination capacity in the brain. And so our thought was, let's reduce these microbial metabolites here. It's going to be pretty tough to alter the microbiome in a predictable way. So let's not start there. Let's start with what the microbes are actually producing and try to remove those. And so we took a very simplistic, conceptually simplistic approach of developing a sequestrant molecule we call AB2004 that actually soaks up or binds to these metabolites as it transits through the gut, which should result in lower levels of these metabolites in circulation. And so we went back to this model that Sarkis highlighted earlier today. We showed that the 4-EPS was actually in the 4-EP producing mice was actually elevated as expected, and the 4-EP negative mice don't produce the metabolite, and that AB2004, which is given orally in the food, was able to reduce the circulating, or in this case, urinary levels of 4-EPS as well. And this actually translated into restoration of wild type level behaviors in anxiety and repetitive behavior. So if you compare the second bar and the fourth bar in each graph, you see that there is a reduction compared to wild type or to 4-EP negative mice. And so this is representative of an anxiety phenotype in the open field. And so administration of AB2004 starts to bring those levels back up to the control levels. Similarly, we see reductions in marble burying as a result of AB2004 treatment, and also reductions in self-grooming, also a measure of anxiety and repetitive behavior, or at least it's believed to be that. So with that, we were very motivated to take this into a clinical trial. And so we went into an open label, keep that in mind, please, safety and tolerability study with AB 2004 in adolescents aged 12 to 17 who were autistic and had GI symptoms. And this was, as I said, an open label dose escalation study in which we gave low dose for 2 weeks. Safety assessment was done. If they were tolerating and everything looked safe, they would progress to the medium dose for 2 weeks, same type of assessments, and then on high dose for 4 weeks. Then we took the drug away, and then we followed up with them 4 weeks later to see what the rebound effect looked like. And so, again, safety and tolerability were the main goals of this study, but we also wanted to see whether the AB 2004 could reduce these metabolite levels and whether we could see any changes in behavior. And since there's always this question of how well mouse models of behavior translate to humans, most would argue not very well. And so rather than pinpoint a particular type of behavioral measurement, we actually measured a very wide range of behaviors in order to give us the best chance of seeing a change if it was there. And so we did the aberrant behavior checklist, we did the Vineland, we did repetitive behavior, pediatric anxiety rating scale, etc. And what we can say is that the drug was very safe and well tolerated at all doses. And we had 2 serious adverse events. Neither was related to the study drug. We had one kid who, before he even got his first dose, was diagnosed with mononucleosis and had to withdraw. We had another kid fall and break his arm. So those count technically as serious adverse events, but clearly not related at all to the drug. Everything else was very mild, transient, and could not be associated with or definitively associated with AB 2004 treatment. So this was actually not surprising to us given the nature of the drug. It's an oral medication mixed with food. The kids eat it, it travels through the GI, and you simply excrete it out in the stool. So we expected a very good safety profile, and that's what we saw. In terms of behavior and metabolite changes, remember that in the mice, we saw a significant anxiety phenotype with exposure to 4-ethylphenyl sulfate, shown here. And we were able to correct that anxiety phenotype in mice with the treatment of AB 2004. So when we looked at the entire cohort here of 26 kids, we saw maybe a slight reduction, but certainly not significant. But I'll just remind you that coming into this study, we did not include or exclude based on a particular anxiety score or any other thing. You just had to have a diagnosis of autism and have GI symptoms. So we had a full range of different profiles in this study. And so what we saw was significant reductions, not only in 4-EPS, but paracresol sulfate, indoxyl sulfate, and several other structurally related metabolites by the action of AB 2004. So this is good evidence of target engagement. And when we actually did a post hoc subgroup analysis of those kids who came in with a high anxiety score, a high anxiety score, so in the pediatric anxiety rating scale, that's a score of 10 or higher, we actually saw a significant improvement in those kids' anxiety scores over the course of the 8-week treatment. We also saw a different pattern of metabolite changes in these kids. And so that gives us maybe the opportunity to go back and do some data mining to understand the relationships of these metabolites, both their starting levels and their changes as they relate to different changes in these types of behavioral scores. What we don't show here is that the reduction in anxiety score persisted through the 4-week recovery period. What's interesting is when we go to irritability, which is a subset of the aberrant behavior checklist, so the so-called ABCI score. We again saw significant reductions in several key metabolites. And at the group level, where we see the very large error bars here, so a lot of variability in the baseline scores, we still saw a significant drop in irritability score. We would do the same kind of post hoc subgroup analysis. We see, again, reductions in the metabolites, and we saw an even greater reduction, more significant reduction in the irritability scores in those kids who entered the study with a high anxiety score, in this case, a score above 15 on the ABCI. So to put this in context, the two drugs that are approved for irritability associated with autism are aripiprazole and risperidone, two atypical antipsychotics. Both have significant side effects that we believe limits their prescription by physicians, because you're trading the irritability profile for a different set of symptoms, such as metabolic disease, weight gain, etc. And so in those studies that got these two drugs approved, they used exactly the same instrument, the Aberrant Behavior Checklist Irritability Score, and their mean improvement was comparable to what we're seeing here in our high irritability group. So what we think here is that we have a potential intervention that acts, it has the same clinical outcome as aripiprazole and risperidone, but without the side effects. And so the interesting thing here is that when we take the drug away, so this is baseline, we see their input score, we see the score reduction at the end of treatment, and 4 weeks later, after the drug is taken away, the irritability scores start to bounce back. What we see here is the metabolite changes in between baseline and end of treatment, and then we see them all go back essentially to baseline by the end of the recovery period. So we give the drug, the score comes down, we take the drug away, metabolites go back up, and the irritability score starts to return to pre-dose levels. So this was encouraging, but again, I'll come back to the open label nature of this study. And we all know that we've seen several studies that are open label, not placebo controlled, that yield very encouraging results. And so we have to be very cautious about over-interpreting these data, but we are very encouraged that we have something that for sure is very safe and well tolerated, and we can actually move the metabolites in a significant way in both blood and urine over an 8-week period. We actually see metabolite levels come down sooner than that. And so metabolite levels bounce right back within 4 weeks of stopping treatment. And we did see significant improvements in irritability and anxiety in those populations who had the high scores coming in. So what this does is it motivates us to do what we're doing now, which is a placebo-controlled, fully double-blinded, randomized clinical trial that is designed to give the litmus test here for the efficacy of AB 2004. And our primary endpoint here is irritability score change at 8 weeks between comparing placebo to high-dose group. And we also have several secondary endpoints that allow us to do analyses of low dose versus placebo, etc. So we will also use the CGI-S, the Clinician's Global Impression of Severity, as a secondary measure as well. And these are all things that FDA is on board with. We have met with FDA. They are, we're aligned on this design. We're aligned on the endpoints. And because the two other drugs that are approved used essentially this same instrument in this same population and a very similar trial design, this takes a lot of the regulatory risk out of the development path for this drug. And let's face it, in autism, there's a checkered history of challenges with getting something through phase 3. We have a clear regulatory path here that we think we can take advantage of. It doesn't mean that ultimately, at the end of the day, this is all the drug is good for. We think it could also help with anxiety, but we're not going to burden our development plan with an anxiety, needing to meet an anxiety endpoint when we have such a strong signal in irritability. And so this trial, which we kicked off a couple of months ago, is actively recruiting. These are the key inclusion and exclusion criteria. So if you have a son or daughter who is in this 13 to 17-year-old range, or you know somebody with a son or daughter in this range who has autism and has challenges with irritability, they may be eligible for this trial. But we obviously have several key exclusion criteria as well. We don't want anything involving antibiotics or anything that's going to alter the microbiome during the trial. Not that we think it's a problem, it's just going to confound the data potentially at the end of the day. We want to keep this trial as simple and clean as possible. So the trial is actively enrolling in the US, Australia, and New Zealand. These stars here are active sites. These are sites where we're activating at various stages of activation. So we have right now about 15 sites up and running. We're going to probably do about 25 sites in total. And you can find more information about this trial at clinicaltrials.gov under this identifier here. So to summarize, we're a clinical stage company that is based on rigorous evaluation and effort in understanding the science of the microbiome gut-brain axis. And we're well grounded in Sarkis Mazmanian's work at Caltech. We've worked very closely with him collaboratively, and again, different from most of the other microbiome companies in that we are developing gut-targeted small molecule therapeutics based on new targets we identify in the gut. And we have a growing pipeline. We talked today about AB 2004 and autism, but we have other work going on in Parkinson's disease and oncology. And last, I would like to thank a lot of people here. I won't go through everyone, but we have the excellent team, highly motivated team at Axial, obviously our collaborators, Sarkis and Brittany at Caltech, the investigators at the clinical trial sites from our phase 1b/2a study. Can't do any of this without funding. We're really happy that we closed a Series C of financing earlier, just a couple of months ago. And one of the key groups we were able to align with and get an investment from was the Autism Impact Fund. And they're already living true to their name, as we've seen here at this conference. So, and lastly, Pramila and your tireless efforts at the Brain Foundation. Thank you very much.
Thank you. That's a great presentation. Go for the questions or
Oh, sure.
There's one question: what is the cohort size for this trial, where the participants also taking antipsychotic drugs at the same time?
Yeah, so the cohort size, we have 3 cohorts: placebo, low dose, and high dose. Each is about 65. So the total N in the study will be just under 200 kids. Right now, we are excluding those taking antipsychotic drugs. We may revisit that in the coming weeks, but for now, we're trying to keep the population as uniform as possible. But so right now, we exclude people taking antipsychotic drugs. So I can't see any other questions, Pramila. Do you see any?
No, that's it. We can move on.
Okay, great.
Please here.
Okay. So next up, I believe, is Jim online? Yes, he is. Okay. So we have, let me see, where did we go? We have Dr. Jim Woody, who is, well, has been around for a while and has done a lot of very amazing things, and currently CEO of Mara Biosystems, but certainly has been part of both large and small companies over the years, and go right back to his days at Centacor, which was really I think, the pioneer in anti-TNF therapy and really set the stage for many other companies to come in and participate in that space. Transformational work, actually, and but we'd love to hear more about Jim's most recent effort at Mara Bio, which is focused on precision medicine diagnosis of autism. Jim, take it away.
Thank you, Dr. Campbell, for the nice introduction. And by the way, congratulations for taking therapy into trials. This is very difficult, as we all know. So persistence pays, and you get a gold star. Let's see if I can pull this.
Well, thank you.
Now, can everyone see this?
Not yet.
I've got to then go back to the share screen. Unfortunately, it just disappeared. I'm going to have to go back and retry.
Okay.
Sorry about that.
So far, we can't see.
It's loading again.
Oh, okay, it's loading. I got it. Okay.
Now I'll try share screen. Can you see it now?
Not yet. Let's see. Here we go. That looks promising.
I think you have shared the entire desktop though.
Do that. How do I get rid of that?
When you hit share screen, you can just select that application, the PowerPoint, or
Not
Right.
sure how.
or you could make it full screen.
Will that work?
If you go to the three buttons at the top left, you should be able to expand to fill up the whole screen. Just increase the size of the window.
Got it. There we go. Sorry about that.
Hey, all right, that's it. All right, good luck.
So first, I want to thank the Brain Foundation for sponsoring the Synchrony meeting. Since I'm a newcomer from the clinical therapeutic field, I've really found your conference to be extremely informative. And a special thanks to Pramila as mission control, who's done an amazing job at herding all of these cats along. So congratulations.
Thank you.
We believe we have a precision medicine company that's aimed at predicting autism before symptoms exist, and ultimately to allow parents a choice for 10 to 25% of autism cases per year, which I will explain later in the next detail. This is the first ever actionable diagnostic test, we feel, for assessing the risk of having an autistic child. And it's really based on the decade-long work of Dr. Judy Vandewater at the UC Davis Mind Institute, who many of you know. We believe it represents a really fundamental breakthrough for a subtype of ASD biology simply by testing the mother's blood for autoimmune antibodies to fetal brain proteins, somewhat akin to the anti-folate receptor antibodies described by Dr. Quantros yesterday. So this is a precision medicine opportunity because we think it's the only technology that can both test for a child's risk of ASD before symptoms exist, and eventually serve as a companion diagnostic to allow parents to decide if they wish the mother to be treated with a novel therapeutic antibody that could eliminate these autoantibodies, allowing parents to have a normal child in 10 to 25% of ASD cases. More on this later with the details. We think there's a sizable market for this simple maternal blood test, which I'll explain as we go along. So I'll spend a moment on our team here. Dr. Marnan Kligfield is chairman of Mara Bio. He was on the board of Cure Autism. now that has merged into Autism Speaks. He has an autistic child and is well versed in all the aspects of ASD care, and is a MiraBio founder and the champion of our technology. Chuck Gardner is also a founder and was actually one of the builders of the UC Davis Mind Institute. He was the founder of Advanced Education and a founder of Hello Housing for ASD, built houses for ASD children. He is currently on the board of Community Living Options, and he has a child with autism and is a especially helpful contributor. Vince Lorenza is former chairman of the board of Becton Dickinson and recently joined our board. He has an autistic family member and is an enormous asset as an advisor on establishing the diagnostic testing strategy for our company. Judy Vandewater, who many of you know, has been working on this for over a decade. She made the amazing discovery and is the scientific founder of MiraBio. She's a real champion. I'm a pediatric immunologist. I was ironically drafted into the Navy from Boston Children's Hospital to manage their radiation injury program, as I had done bone marrow transplant. My Navy group was the first one to determine what HLA matching was necessary for successful marrow transplant. And my colleagues and I, with congressional approval, founded the National Marrow Donor Program, which has actually helped over 100,000 people receive transplants. I moved on to be Chief Scientific Officer of Centocor, where my team developed, as they noted, the first anti-TNF inhibitor called infliximab or Remicade for rheumatoid arthritis, Crohn's disease, and psoriasis, which is still sold by J&J for over $1 billion a year. I moved on and became general manager of Roche Bioscience, which is formerly Syntex, for 8 years. So I've recycled several times. I went on to be founding CEO of Oncomed, a therapeutic antibody company. And then I joined a venture fund, went over to the dark side and invested in 10 to 15 companies. 2 of them were antibody diagnostic technologies. We founded several companies that made drugs bought by big pharma. And one of them is actually selling for over $1 billion a year. So I've done a lot of different things as we moved along. These are our key opinion leaders, some shared with colleagues presenting at this meeting, who have provided superb advice to us for how we manage the development of this test. And the developmental specialists, we have Dan Corey, who I'm sure many of you know from Nationwide Children's, Robin Hansen from UC Davis Mind Institute, and Tom Fraser, the former CSO of Autism Speaks, all are phenomenal advisors for us. We also have David Traver, a very insightful local pediatrician here in Palo Alto that has a huge ASD practice, even a prenatal practice in his pediatric group. We also have OB and IVF specialists like Mary Poland, who ran the Stanford IVF clinic, and Sandra Carlson, who runs the Yale IVF program, and Jan Frudford, who has a large OB practice here in the area with ASD families. We have Bobby Rethmiller to help us design clinical trials, and Stan Lapidus is an advisor. Stan had a former company, Synaptics, for ASD genetic diagnosis. He also invented the Pap smear and Cologuard. So he's an amazing person with Sage device. Lastly, we have Nina Agenofor, an expert in diagnostic trials, statistical analysis, AI, and machine learning from Stanford University, who's really validated our technology. Our internal team is made up of several experts in their field as we convert this laboratory test to a commercially available test, which is a real challenge. Mags McInerney is an expert in analytical development for both CLIA and FDA IVD diagnostics. John Stinsky was actually part of the initial PCR team at CEDAS. He became CSO at CareDx and was at Quest Diagnostics and Roche Molecular. He has a wealth of experience and an amazing person to have help us. Catherine Tynan has commercialized a whole range of tests, both in the CLIA area and in the FDA-approved in vitro diagnostic space. Nikki Zoll and Kathy Nichols are helping us with marketing. And Deborah Shuren, with the huge amount of experience, as our Chief Financial Officer. And Michael Paul has recently joined us. Michael was the founder and CEO of Lineagen, a very successful ASD genetics diagnosis company. There's nothing that we're doing that Michael hasn't already done, so he's a tremendous advisor for us. So we've heard several times now the new CDC estimate of 1 in 44. We also see this as increasing on a worldwide basis, not just in the US. And as Dr. John Slattery mentioned yesterday, it's increased 241% in the past 2 decades. We have got worldwide rights for our test, and so you'll hear more about that later. Because there are really limited options to determine the risk before symptoms exist for ASD, and a delayed diagnosis really leads to delayed access to the critical interventional therapy that we're all aware of. There's a unique challenge to getting children into the earliest intervention. The behavior-based tools are not really reliable until the child is about 2. Average age, as you've heard from several of the prior speakers, is about 4, because there's a large backlog in developmental pediatricians and psychiatrists and psychologists to examine and begin treatment of these children. There's also a lack of consensus about who should actually be referred and when. Our KOLs tell us if a child has missed a developmental milestone, as say a positive MCHAT, or the parents are concerned, they would actually order our test, and if positive, would move the child to the head of the line for the clinical evaluation and earliest interventions to give the child the best earliest chance. We feel that technology could conceivably decrease the time to intervention by 12 to 36 months with a reduced lifetime cost and benefit for not just the child, but the family and the community as well. We all know that the window of opportunity in these child's lives is small, where we can achieve the best outcomes. So we at Mirabai are trying to get them into therapy as early as possible. If even before symptoms, parents can plan a proactive therapy for them. So Judy has developed some very innovative science behind this biology. I'll spend a few moments on this graphic view of how this happens. Some women, for unknown reasons, have preformed antibodies to infant brain proteins that they carry for years before and after pregnancy. At the time of pregnancy, these antibodies cross the placenta at about day 100 and damage the infant brain. You can see from the brain pathology slide on the right, from the animal that has autistic behaviors induced by immunization with these MERA proteins, that the neurites are blunted. So the brain's electrical system is not well connected. I was very interested in Dr. Lipton's comments about his MEF2 activator and neurite outgrowth. Dr. Vandewater has published over 15 peer-reviewed publications that support the findings. The most recent was very compelling. We'd be happy to send you a copy. As we use our Stanford statistician to validate the findings that we had made. The test is very highly predictive. There are very few false positive, and we have a proprietary position. What convinced me to join this effort is seen here, and that was that passive MERA antibodies given to primates resulted in offspring with autistic behaviors. This is an extremely compelling proof of concept in a primate. I doubt if any other technologies have anything similar in proving the theory of their test. Here are the 8 specific proteins. It's noteworthy that these brain proteins really exist in fetal humans, primates, and rodents, and they're largely downregulated in adults. So Dr. Vandewater has developed animal models where the offspring of animals immunized with these MERA proteins demonstrate antisocial autistic-like behavior. The videos of these animals is very gripping. This immunopathology slide just shows that the antibodies do actually enter the brain tissue of these animals that are immunized. Now I'll spend time for some clinical correlation data. The test is a standard ELISA test. We've done 2 retrospective and 2 prospective studies. Others are being performed to date. So here is some of the data. The 2 prospective studies, as outlined, involved analyzing samples in a blinded manner, then calculated the accuracy of predicting an ASD outcome, which was very high as seen, almost over 99%. We use this for validating the laboratory developed test. The 2 prospective tests used samples drawn during pregnancy for other reasons. Then the patient was followed for fetal outcome. Again, the test predicted ASD outcome very accurately in over 95% of the cases. With 700 samples analyzed, the data really supports the MERA hypothesis with real biology versus algorithmic associations. We initially planned to use the test to get children to the earliest beneficial therapy. Other follow-on indications will be for women with a prior ASD child wishing to become pregnant today. Again, if she has the antibodies, she'll have another autistic child. Women over 35 contemplating their first pregnancy, or women in IVF programs. Our OB-IVF consultants strongly support these particular indications. Now, we will not be testing any pregnant women. Now let's move on to the therapy potential for this. This is the next generation of things coming along. So there's a new technology called anti-FcRn antibodies just being approved for therapy in various autoimmune conditions like myasthenia gravis, where the patient has autoantibodies to their own acetylcholine receptor, which causes severe weakness. The 5 current companies that have these antibodies in trials are seen on the right here. I think 1 or more of them will get FDA approval probably this year or early next year. When anti-FcRn antibodies are administered, as shown on the graph on the left, serum IgG antibodies can basically be eliminated. Momenta, purchased by J&J, was actually treated pregnant women who had antibodies to infant red cells, where the first baby failed to survive. The therapy allowed a normal child to be delivered. The rest of the trial is still in progress, but the proof of concept that this can be done is clear and safely. The science behind this has to do with how the body processes circulating IgG antibodies. The IgG binds the Fc receptors on various endothelial cells. If the antibody is damaged, it immediately goes to the proteasome and is converted into its amino acid. If not, it's recycled back into the circulation. If the FcRn is blocked, then all of the antibodies are degraded. The FcRn is actually also the same receptor that allows antibodies to cross the placenta to the baby. So anti-FcRn actually blocks both. MERA Bios filed intellectual property on the use of anti-FcRn for MERA indication. Shown here is a rodent that's been immunized with MERA proteins and has antibodies to CRIMP1. We reduced the CRIMP1 antibodies with a single dose of anti-FcRn as a proof of concept that this can be done at least in an animal model. Let's look at our timelines and market size for this particular test. So we are partway through analytical validation. We'll be doing a clinical validation study in parallel. The anti-FcRn program will need quite a lot of animal model proof of concepts, which will probably take 12 to 18 months or more to do. The potential market size for the MERA test is large and growing. You can see the numbers, almost 750,000 people might be eligible for the test. So it's a very large market. So we think our test represents a paradigm shift. It's a simple maternal blood test, provides accurate risk for having a child diagnosed with ASD even before symptoms exist, gives us the earliest risk assessment and the earliest best therapy for the children with the lower cost, providing clinically actionable information for clinicians and choice for parents. We have a very proprietary position, large serviceable market, low risk regulatory business model, a CLIA regulated test not requiring FDA. We have a number of exit opportunities with different groups. And for the larger vision These proteins have genes, RNA, pathways, and epigenetic influence, and may finally allow the vast genetics database to correlate with real biology that we can show damages the brain, and maybe give us new aspects to ASD, including potential therapies. So I will stop there, and I'm happy to answer any questions you might have.
Okay, thank you very much, Dr. Woody. And so there are a couple of questions showing up here that I can see. I think we have time for maybe one or two. And a compromised blood-brain barrier. And this doesn't seem to be an issue in your case. Is that right? You just showed some data where it does actually get into the brain. Is that right?
Yeah. In the developing infant, I think the blood-brain barrier is less secure than in adults. That was also outlined by the antibodies to the so similar sort of things that they do leak across into the brain. Interestingly enough, we find these antibodies in blood bank samples. So the antibodies are quite widespread, not just in the women that we're considering.
Okay, thank you. And then there are two related questions here about the prevalence and pattern of these antibodies in women. So does the antibody profile of a woman change over time? That's the first question. And the second is, what % of women with an ASD child have these antibodies?
I'll answer the first question first. Depending on the geographical population, we're seeing anywhere between 10 and 25% of the women have these antibodies. Average is about 20%. And different groups have different sets, since there's 8 of them here. We do have some initial evidence that Dr. Vandewater developed, may want to comment on it since she's here, that antibodies to CRIMP1 lead to the more severe or maybe even the profound cases of autism. That's something we're going to try to define in the next year with a very large clinical validation study.
Okay. And I see that Judy Vandewater has her hand raised, it looks like. Do you have this? I don't know that she can get into asking a question live, but maybe if she could put it in the chat, that would be great. So maybe in the interest of time, Dr. Woody, you could come back to the Q&A. Oh, sorry.
Yep, she let me in.
Okay, great, go ahead.
So I think to answer the question of do they change over time, which is an excellent question. That is actually one of the goals of the MARBLE study. We have longitudinal samples in those women, and we're seeing about 25% of those women. These are women who already had a child with autism, and we're following them throughout an additional pregnancy. And one of the things we are seeing is there may be some slight changes over time. And so that's something that we're really interested in, is how those change over time and what that might mean when that's happening during gestation.
Great, thank you, Judy. That's great context there. So maybe there are a couple of other questions, both in the Q&A and the chat, that maybe you could come back to, Jim, while we proceed to the next talk.
Great.
So without further ado, I'll move on to the next speaker, who is Dr. Eric Wan from WAVE Neuroscience. He is both president and chief medical officer there. And Dr. Wan has a very decorated career, both in the military and also in the general population as a physician, and has seen, I think, the full spectrum of different types of medical situations, and now he's applying all that experience and expertise to developing TMS, or transcranial magnetic stimulation for Kleefstra syndrome. And maybe I'll just turn it over to him now to tell us about what's going on at WAVE Neuro.
Sure. Thank you so much for the introduction, and we're really honored to be with all of you today. I've had the opportunity to listen to a bunch of the presentations throughout the last 2 days, and it's really inspiring to see all the advances in the field. And we'll share a bit about ourselves today, but just a virtual standing ovation to Pramila and the Brain Foundation. This has been an exceptional symposium. And we'll share some of our contributions to the science. I'll start by introducing our disclosures. Alex, who's to my right, and myself are employees and minority shareholders of Wade Neuroscience, but we will just present the science and the data and avoid commercial bias. We have numerous funding resources, which we'll touch on in a moment. And then Dr. Mason Harrell will also present his experience. He has a daughter with Kleefscher syndrome. This is an autistic, a genetic form of autism with some unique characteristics. He has a unique perspective as well as both a physician, a scientist who's triple board certified. So we'll launch right into it. Part of the reason you're seeing this intermingling of military approaches, special operations, and then autism, it left me scratching my head at first glance, but this technology was really engineered and innovated to help the brain function better and more efficiently as an organ. And when we use that as our end goal, we can touch many different conditions and diagnoses. We're very careful to say this is not a panacea or a cure for any of these conditions. It's not even disease modifying, but we're seeing some significant and promising data as it relates to a number of different symptom clusters, and as it relates to developmental delay, we're seeing some encouraging results. So the military's interest is in human performance and optimization, and this can be applied across a number of different platforms. And so we have a wonderful team, proud to work with all these individuals, scientists, engineers, physicians, all getting together with a common goal and purpose. We also have a deep bench of advisors and consultants, some that you may recognize, but all both wonderful people who are purpose-driven and have really advanced the science for us. So feel very privileged to be working with all of them.
Hey, excuse me, Eric, I don't think your slides are changing. I'm not seeing them change here. I'm still seeing the title slide. Are you in slideshow mode?
I am, but let me try redoing that. Hopefully this will work. Oh.
When you share your screen, share on as well.
That was working there, Eric.
All right, let me try that again.
Go and
The try before work. Oh, there we go. It's good.
Thank you.
Sure. So this is a bit about our platform. This is our team, and here are our consultants. And so I know we ran through those relatively quickly. We can make these slides available if people are interested in reviewing in more depth. But in the interest of time, I've been advised we're running a bit behind. This is our process. We first get a brainwave recording. Many of you may be familiar with quantitative EEG. It's a brain mapping technology. Alex is going to speak to that in much greater length momentarily. We run this through neurocomputational analytics and a normative database and come up with a report. This has been transformed from what used to be a 30-page report into a one-pager that is intended to be much easier to understand, taking some very complex neuroscience and distilling it into more easily interpretable biometrics. And then we're stimulating the brain using an FDA-grade class 2 medical device. It's called transcranial magnetic stimulation, but we're individualizing and personalizing protocols unique to each individual. And then we can create a virtuous loop of doing these EEG scans every couple of weeks, and then ensuring that we're achieving the progress objectively and scientifically. We're very proud of our research partners. We've invested quite a bit of resources, time, and energy into validating the results that we're seeing. This is a partial list, but we're embarking on even more studies in 2022, moving to larger sample sizes and outside of just pilot data. And I'll turn it over to Alex to talk a bit more about our EEG science relative to autism.
All right, so heading over to what Dr. Ron just covered, an EEG is a measurement of cortical electrical activity. It could be taken during eyes open or eyes closed states. It could be taken when someone's awake or asleep. And there are different frequencies of activity that are generally expected during these different stages of alertness or sleep, eyes open, eyes closed. And when you start looking at subjects who have been diagnosed with autism or developmental delay, developmental disorders, there are common endophenotypes or features that reveal disconnective features in this EEG. So there are features, as you can see here on the left. This is looking at default mode network function, which is generally a reflective network and occurs during eyes closed waking states. We see when we compare this as well against normative controls that there are differences. And I'm going to focus on that on the next slide as well. Here's a comparison. On the left is typically developing controls versus autism confirmed children. And you can see that there are these darker lines that progress from left to right of the screen, going from 30, 60, 90 to 120 months up to 10 years of age. And we can see that the x-axis is frequency for eyes closed, resting EEG. And everyone starts with a frequency approximately below 8, but you can see typically developing children around 30 months have a dominant brainwave frequency of about 8 hertz or 8 oscillations or brainwave oscillations per second. That progresses up to about 10 to 11 by about 90 to 120 months. And when we're comparing that against autism, what we can see here is that frequency maxes out at about 9 to 10 hertz. So there's a frequency component here that's intrinsically underdeveloped when we're looking at children with ASD. And this component is something that's important to recognize and understand, because when it comes to alpha activity, this has been demonstrated to be reflective of our sampling frequency, essentially the number of pieces of information we're taking per second. It's explained by Nyquist's rule of frequency sampling, but the faster the frequency, the faster generally our sensory perceptive rhythm is. So this is one of the components that we find is generally reported in literature as disrupted in autism. Another component is something called coherence. And coherence is how well connected the brain is, given the different frequency bands I referred to earlier. You have slower frequency activities, theta and delta, and these generally occur during stage 1 sleep or deeper periods of sleep, and then waking rhythms, eyes closed being this alpha rhythm, and then eyes open being beta. And fairly reliably, what's found is that this alpha rhythm, in addition to being slower in its expected frequency, it's also less connected in general, especially between anterior and posterior cortical regions. That is, it tends to be hyperconnected posteriorly, and there's underconnectivity between posterior and anterior regions. So you might have excess of alpha activity in posterior regions of eyes closed resting EEG, but you'll see that it's quite underdeveloped in frontocentral regions. This has been confirmed and reconfirmed with multiple studies. Here's a connectome. On the right, you can see the EEG locations. The posterior locations are on the right side near the bottom, and the anterior locations are up top near the left. And red are areas that are overconnected, and blue are areas that are underconnected. You can see that there's, as a consequence, underconnectivity frontally and overconnectivity posteriorly. So this is something that's been well recognized and published in literature, and it corresponds well with the symptom profiles that are generally observed in ASD. So we take advantage of this insight, and for every subject, every patient who comes through, we take a look at their EEG and try to reduce this various information, which can be very overwhelming to someone who doesn't have any background in EEG, and simplify it to a picture here. And you can see this picture has posterior, central, and frontal labeled on it. And we have something called the Fourier transform here, where on the left side of the screen would be the raw signal, and we're simulating it here in frontal and posterior regions. If we count the number of oscillations per second, and let's say there are 10 oscillations per second, then we'll be able to see on our FFT a peak at 10. Similarly, if there are 9 oscillations per second, our FFT will demonstrate a peak at 9. If there are 12, a peak at 12. If both 9 and 12, a peak at both 9 and 12. So we can get from this a frequency component and also an amplitude component for the EEG duration that we're recording. And we tend to see this for 10-minute eyes closed EEGs. And what we see on the right is for typically developing adult or child, a dominant posterior rhythm at about 10 to 11 hertz. You can see that peak there. And then the same corresponding frequency in central and frontal regions. The peak is sharp, which tells us that there's no other activities occurring. We don't see peaks slower off to the left or faster and off to the right, neither theta or beta frequency ranges. Now, when we take this test and we start looking at our children who might be coming in with ASD diagnoses, we start to notice fairly profound changes or differences rather than just resting eyes closed function. Here we see highlighted in red on the right, the alpha frequency band for a child with autism. We can see that there's a frequency of about, let's say 9.5 hertz in posterior. And we look at frontal and central, there's a corresponding alpha activity at about 10, but it's not the largest EEG rhythm in the FFT spectrum. We can see there's also activity off to the left in this theta range. And as a consequence, the amplitude of alpha activity in the frontal lobe could preferably be higher. So we take this information and we then utilize it with neuromodulation in order to try to get the EEG that's on the right to shift and look more like the EEG on the left through entrainment of cortical tissue. So the technology referred to earlier, this RTMS, we call it magnetic EEG, EKG resonant therapy. We calculate this biometric frequency from this FFT and then provide that frequency of stimulation into cortical tissue corresponding to the frequency that we're visualizing with this FFT tool. So I'm going to hand it back to Dr. Wan in order to look through a couple of cases.
Thanks, Alex. So this is a case that's kind of near and dear to my heart. It was one of my introductions to the technology. At the time I came across this, I was actually at the Boeing company, most notable Fortune 50 aerospace company. I was playing a role as their chief physician and a secondary role as their technology officer for health services. And one of my neighbor's children who had a fairly significant autism had shared with me a bit about the technology. And you can see some of the history here. At that time, a 6-year-old male who was born prematurely. There's some more details there. And up until 18 months, was really thriving, met all of his milestones, was babbling, crawling, used words. And then around 18 months, there were some lost milestones, and he stopped responding to his name. He lost his words and language, began stemming, doing hand flapping. All of his tests actually for quite a while was coming back normal. His pediatrician had actually provided quite a bit of reassurance, but over time they started him in ABA therapy and speech therapy until he eventually came in for treatment. And this is his EEG upon arrival. And you can see, as Allison mentioned, there is a deficit, if not absence of alpha activity in the frontal and central regions, but the peak in the posterior region where there's hyperconnectivity. And this morning I heard some talks about the MEF2C gene and imbalance of excitatory and inhibitory neuron development. It actually got me really interested because we're seeing some convergence of scientific disciplines here. And if we're of the belief, as Alex and I are, that science always converges on the truth, a lot of these dots are really starting to connect for us. But this EEG was taken 2 weeks after treatment, and you can start to see emerging some alpha peaks in the frontal and central regions. And that was corroborated by many of his behavioral symptoms, the behaviors that had improved, in some cases quite profoundly over time. And so we could see within 2 weeks, he was calmer, was starting to improve his comprehension. He was starting to imitate the children around him instead of Aunt's name for the first time. And then fortunately, as with many of our patients, he came back in a year, and we're very encouraged to see that he continued to make progress. And it's important to note, this progress was made without treatment. It seems to us that once we start to activate these neurons and those neural networks start to light up, the body does much of the work for us. And this is true to, I think, many of our medical paradigms, that we tend to be born and engineered as healing organisms. If you put two ends of a wound marching together, it heals. Two ends of a broken bone together, it'll heal. It seems to us that with brain activity, if you move it in the right direction, the trajectory of that improvement tends to improve with time as well. So this child came back for treatment several years in a row. Hopefully this video will include audio, but this is a before and after video of this child that the family's given me consent to share this. Hopefully this will play.
And then they had to go, they went on vacation, they went somewhere. And if we do miss one, or if he moves too much, we usually just add another one to the end.
Oh, that's not a T, it's a D.
Kid.
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 Kid. Kid. Good boy.
Rat. Good boy. Good boy.
When?
So before I turn it over to Alice, I should mention not every child has this kind of progress, but we're really encouraged by his change, and it was part of what compelled me to move over to join this organization. And he is now much older, obviously, but he no longer requires IEPs. He's at grade level and is making friends and has done quite well. So let me turn it over to Alice to share a bit more about some of the work that we've done.
So I'll show another case report right here, and we're going to go ahead and launch into some of the chart reviews that have been performed. I just wanted to note that for the therapy, we measure individual alpha frequencies, which can vary person to person. So we task the RTMS device to match with the frequency that we're measuring in the EEG, and one person might receive stimulation 9 hertz or 9 times per second, or another one might receive it 11 times per second. And these are daily 30-minute sessions where stimulation is applied over the course of Monday through Friday, generally 5 days a week, and then EEGs are taken serially, as was mentioned earlier. Here are twins, identical twins, and you can see this line here, February 15, February 17. This is the baseline for the identical twins, and their EEGs look a little different. And the severity of their disorder was also similarly different. And we can see from left to right the general and gradual formation of a peak for the top at about 10.5 hertz, and for the bottom case from left to right, a gradual development in this rhythm also at about 10. Although this case on the bottom far right has a peak also posteriorly at about 8. So when it comes to cortical tissue and network function, it's different person to person. But following 1 month of therapy, for both cases, they reported improvements in cognition. Attention to language was very exciting for the family. So we've been seeing this for many years now, application of the technology. And there are a couple of studies and chart reviews that have been performed on the populations that have gone through this. This was one of the first ones back in 2014, an N of 26 that followed up children went through clinic that received this for mean of 24-month period of time after the period when they received and underwent the therapy. And it was 26 cases who had a baseline CARS score, childhood autism rating scale. I believe this audience understands that score. Severe is, I believe, 40. 40 plus is where we get moderate, and severe is 40. It's a bit higher. But started off with the CARS score of 41, and by the end, at the end of 2 years, had an average CARS improvement of a reduction in 11.7 points, which was quite profound and impactful. To note, the cutoff score for some of these, some perspectives is between 28 and 30 for CARS. But this is just the initial look of in-clinic intervention. We more recently took a look at subjects who had received 40 sessions over the course of 2 months of consistent therapy. And you can see here baseline CAR score. This is an N of 167 taken from a couple of clinics who were providing this therapy. A baseline score here of about 39.1. And then over the course of the 2 months, quite an impressive reduction down to an average or mean of 33.9. We went ahead and further stratified by baseline severity just to take a look and see, who would be most benefiting from this type of intervention, at least in this clinical setting. And what we were excited to see is those who were most severe had the greatest proportion of reduction, and those who were near to the bottom, they didn't improve as much. And we can see from this that symptom severity was in some way corresponded to the degree of change over the course of time. This technology we're seeing applied not only in the States, but in other countries. An Australia group recently went through, and this is in progress for publication right here, and looked at 12 subjects as they received stimulation over the course of approximately a month, and they found BCS and PCS before and after stimulation that there was quite a few domains on the CARs. You can see the subdomains on the right that changed dramatically. Adaptation to change being one of them, and the taste, smell, touch, use, and response, and then fear and nervousness. So there were a couple of domains that were more responsive, at least, but we're excited to see that this is having an impact in multiple domains, and not only on populations in the States. And just to kind of bring back to what we're seeing on the scientific level, we're trying to interact with and provide neuromodulation to cortical networks. And autism is essentially thought of as disorders, a spectrum disorder, where there may be different types of real diagnosis hidden in there. And just to highlight that, Phelan-McDermott and Kleefstra syndrome are also considered to be subtypes of autism, or have a population percentage that also have autism. We're finding and seeing that there's a similar change in networks when we're providing neuromodulation to these networks and to these subgroups. I'm going to hand it over to Mason Harrell, who has a pointed discussion on Kleefstra syndrome. Mason, if you're there, go ahead.
Yep. Thank you so much. Everybody can hear me okay? Yep. Good. Yep. Yeah. So I'm a physician, as was mentioned before, and as a physician, have a little bit different perspective and experience with a child with severe disabilities and autism. And I really appreciate being able to be invited just to speak for just a couple of minutes here to all of you. And I appreciate all of your work that you all are doing in this field. It affects our lives in ways that all of you know or can imagine. So Eleanor, our 3.5-year-old today, we found out she had Kleefstra syndrome at age 1. She is the light of my life, and as you can see, that smile, yeah, it can stop you in your tracks. So the best thing that I can say, probably in my experience as a scientist and as a skeptic in general, is that Eleanor went through a period of time where she really started a new trajectory of progression, of development. And that's the first time that happened. That's when she started walking. When she started walking, there was just a brain developmental increase in trajectory. And the second time that happened was when she went through transcranial magnetic stimulation. And it wasn't, within a first week. It was actually probably around the 4-week, 6-week, 8-week mark, where not just us, we started noticing it, but our therapists were blinded. They didn't know this was going on. And they started saying, whoa, something's changing in Eleanor. And it was really exciting. And it was just like when she started walking, where the trajectory changed. Specifically, we saw that, or we experienced that Eleanor's cognition speed, just the how fast she could process information, made a significant increase. She was speaking back then. A dog was a d, and that was it. And during that time, she started closing words. So she went to say dog, and instead of d for dad, it was dad. Really neat. And then the other one was attention. She was just paying. She started paying attention more. And the second thing I wanted to say, I appreciate, Eric, what you said earlier, where the effects lasted longer. It's been a while, Eric, and we moved about a month ago, and I didn't have a chance to update you, Eric, but she continues to progress, and now she's on to 2 to 3 words, and 4 syllables. So the progression has continued. In her since the treatment, and she finished around the treatment for the first time. She finished in April, and so that wasn't too long ago. And that's all I have. Yeah.
Great, thank you, Mason. It's a little emotional. Mason's a friend, and his daughter's just a beautiful, wonderful child, and so to see these kind of changes is tremendous. But we want to share a bit of the science, the background science that we were able to observe. And so I'll turn it over to Alice to talk about her EEGs as well.
So this is, thank you, Mason.
Yeah, thank you so much. I held back my emotions, Eric. I did a good job, I thought. Being a scientist, being a doctor.
So this was the baseline EEG. You can see here that there's a peak posteriorly at about 4, and one under 8 at about 7.5. We're finding with some developmental delay cases that they don't develop alpha activity. If the frequency doesn't even emerge past 8 hertz, and that's something just to keep in mind for this case. But we can see similarly same frequencies, 4 and 8, although they're quite small in this EEG at baseline. And Eleanor went ahead and went through 2 months, I believe, of this daily stimulation. And we can see that this is after 1 month. We took the baseline EEG and we waited before commencing therapy until about June, mid-May, June. So this is after 1 month here, and this is after approximately 2. And we can see that from baseline to more recent EEGs, that there's been quite a large emergence of both rhythms. Prominent 4 hertz is quite high following the intervention therapy, but we can also see 8 hertz or 7.5 starting to emerge. There's no alpha rhythm that's emerged yet, and that's not uncommon as we see in some of these delays. However, the fact that we have such a large increase in cortical activity at the scalp certainly points towards possible mechanisms, including long-term potentiation, improvement in cortical network synchrony, and all of these other domain factors that are normally disrupted in the autism spectrum domain of EEG and cortical functional signatures. So just with that, it's so endearing to be able to work with this case. And Mason, thank you for being on. We're going to go ahead and open it up for questions and discussion.
But before we open up for dialogue, I just want to share that the story's not quite over yet. Mason is just love of a father and the momentum from this case. We've actually gotten IRB approval to do a small trial. Cleavage is exquisitely rare. It's a chromosome 9 deletion, and so getting recruitment for these types of studies is a bit challenging. But the Cleavage, the National Center of Excellence, is at Boston Children's Hospital, and Dr. Alice Colazon will be the principal investigator. We've designed a trial. We're currently rallying funding to do that trial, and we're hoping that'll launch in 2022. We're also looking to build upon our pilot data and to do a multi-center large pivotal trial for autism spectrum disorder. So we're really working hard to advance the science, and just honored to be here with all of you as part of this discussion. And from there, we can open it up to discussion.
Thanks, gentlemen, and congratulations. I mean, it's really remarkable what you just showed, and if that's predictive of where this is headed, it's going to be it could be really, really significant. So best wishes on that. A couple of questions here really quick before we move to the final presentation. One is, how do you handle doing EEG with your eyes closed, quiet, sort of with an autism population, and do you sedate them? Just trying to pick, there's a question around how that operationally works.
Yeah, I'll go ahead and take that. There's no sedation. The technicians who are doing this have been trained and gone through a vetted process that requires hours and hours and hours of training. It's very difficult to put a cap on the head of a child with autism to get them to close their eyes, where you have to use cotton swabs. There's a lot of patience involved, and the EEGs themselves can take multiple sessions before we get something that's clean. But they work cooperatively with the family. They send the caps home with the parents in order to habituate the children to the test. There's a lot of effort required in order to get something that's clean. And even if we're unable to get something that's clean, the family may be informed that the visualization on the data might be a little bit less ideal, but you can still calculate a method for therapy regardless. So, that's closely handled with the family and with an excellent team of EEG specialists in all the areas where this is being practiced. Great, thanks. And sounds analogous to some of the challenges that we've seen in getting good MRI data in kids with autism. But one question I had was, so far what you showed was this treatment as a standalone, and the response times are pretty quick. So would you expect maybe even either more pronounced effect or no synergy between that and, say, ABA going on at the same time, or other intervention going on at the same time? And I guess embedded in that is, how would you do those trials? Maybe that's a longer term question that we could take offline.
Well, I would say we strongly encourage families to continue with ABA therapy. We view this as complementary. It's very much a team approach. And we've seen tremendous progress with families who do ABA, and we view this as kind of synergistic. How we tease that out in a clinical trial, that is a bit of a challenge. We want to see in isolation what the therapeutic benefit, the safety, and the efficacy of this treatment would be in isolation. But in practice, our oath and our mission, of course, is to serve our patients in the best manner possible. And we believe that the multimodal team approach is going to be the best for each family. Mason, I don't know if you have any thoughts on that either, in terms of Eleanor's approach.
Yeah, absolutely. Yeah, we kept Eleanor in all therapies at all times during the treatment, and coming from I won't say anything else. Yes, and yes.
Okay, great. So again, thanks, bravo, guys, and so she is Chief Scientific Officer of Gillicolite Corporation. She's also a longstanding practicing pediatric psychologist with a PhD in cognitive psychology from Princeton and postdoctoral work at Harvard. Really originally focused on language acquisition and processing, but really curious to hear about the TPM work that you're going to talk to us today. And I'll get out of your way now and let you take over.
Thank you so much for the introduction. It's a great privilege to be here, and I'm going to share my screen. Okay, I hope everybody can see it. So thank you to the Brain Foundation for inviting us to speak. I'm speaking today here on behalf of Gillicolite. I'll be talking about our first clinical trial that we have just completed. And before I move on to the presentation, I just want to give credit to people who deserve it. These are our co-authors, our scientific advisors, people without whom this research wouldn't be possible. Special thanks to Dr. Fratkin, who is a pediatric psychiatrist and who acted as a PI on this clinical trial. Many thanks to Professor Nazir, who shared her wisdom with us in the most generous way. without all these people, this research wouldn't be possible. So with that, I'll just move on. I have to say, I'm so lucky to present the last. I feel like I can skip through many slides of the introduction or give just really high level of introduction. What is transcranial photobiomodulation? In some ways, like transcranial magnetic stimulation that was described before me, it's a way to stimulate brain. This is what Professor Navier called optodynamic therapy. And maybe it's the most accurate name for this technology. But basically, it's a non-invasive treatment, and it stimulates brain transcranially with near-infrared light. In our trial, it was pulsed near-infrared light. Now, what happens when near-infrared light reaches the brain? It's being absorbed by the cells. Specifically, it activates mitochondria. And there were so many excellent presentations, amazing presentations at this conference that showed intimate connection between mitochondria and autism. Basically, there is almost always some kind of mitochondria dysfunction, if not a full-blown disease or disorder and dysfunction present, which is underlying autism. So with this stimulation, we activate mitochondria to produce more ATP, which results in brain healing itself, like was so profoundly said before, body, brain wants to heal, and with a little bit of help, it will. It promotes synaptogenesis, it promotes neurogenesis, and it reduces brain inflammation. Oh, it also, like I said, it promotes synaptogenesis, so it increases brain connectivity, which is one of the underlying causes of autism. This technology has been studied in many studies that looked at dementia, stroke, traumatic brain injury, and depression. And it has been shown to be extremely helpful. For example, in aphasic patients who suffer from stroke, it has helped with language production. It also has been shown to be helpful with memory, mood, sleep. Why autism? It's actually, I feel that this technology was intended for autism, always. First of all, it improves functional brain connectivity, and I'm very thankful to Nero Wave, who talked about connectivity before me, so I don't have to go again and explain what it is. It also affects brain oscillations, and again, just like presenters before me, I'll be talking about brain waves as one of the possible biomarkers of autism. Why look at kids with this technology? I think, again, this technology is suited for children because the skull is not thick. I think it's about 3-4 millimeters thick only in young children. So the penetration of the light is not an issue. There is a lot of synaptogenesis still happening because the children are so young. There is neuroplasticity. Again, with a little bit of help with brain stimulation, the brain will heal, and the connections that need to form will form. So I think this brings us specifically to our clinical study. I just want to show you a little bit of the data. I believe it was already presented yesterday by Professor Siranoglu, but still, this is a study that used this technology, transcranial photobiomodulation, in 2018. Dr. Leisman and his colleagues used stimulated occipital lobe with near-infrared light, and by week 4, they found a major drop in symptoms as measured by clinical global impression severity scale, and this reduction in symptoms sustained until week 8, which was the end of the trial. By the way, they did a follow-up. They followed these children, and children were from 5 to 18 years old. They followed them 6 months later, and the gains that were made during this trial remained. So children who improved continued improving. This is a study that was done in Massachusetts General Hospital with adult participants by Professor Siranoglu and Professor Kasanam. They also used transcranial photobiomodulation. They stimulated different brain areas. I believe they stimulated F1 and F2. And they also measured improvement with clinical global impression severity scale. There is no control condition in this study, but it produced very hopeful results with adults in that the symptoms did decrease by the end of the study. So by the midpoint, which was week 4, there was initial significant decrease, and it continued by the week 8. So we do have some preliminary data, and now we wanted to look at EEG specifically. Why EEG? Why is this endpoint so interesting? For autism, the importance of EEG has been already discussed before us, so I could drop that slide. But for transcranial photobiomodulation, there was a study reported in 2019 by Zamarodi and colleagues. They looked at healthy adults and they applied transcranial photobiomodulation for 20 minutes only. And after this single application, they detected the reduction of slow frequency waves, which, because these were healthy adults, the slowest frequency was theta waves, and they detected statistically significant reduction in theta waves. So from that alone, we knew that brain waves are receptive to this, sensitive to this kind of stimulation. We could look at them. And so that brings us, without further ado, to our first clinical trial that we have conducted. I'm extremely proud. Our company, Jellicoe Light, has conducted the first FDA-regulated clinical trial on this technology. It's an IRB-approved trial. We recruited children 2 to 6 years old, and the study was conducted in clinics in Brooklyn and Manhattan, and we did our best to recruit children from diverse background. We recruited male or female participants between years 2 to 6 years of age. We really closed. We received so many calls. Parents are so desperate from 8 and 9 years old, and we said, no, we're stopping at 6. This is our first study, and we really want to get into that critical window for language learning, because we want to see what this way of neurostimulation does, what it does, how it affects language learning. All our participants have been already diagnosed by moderate or severe autism spectrum disorder, not by us, by a licensed professional. And we allowed all participants to continue receiving the therapy. If somebody was receiving ABA, of course, please do. Our only request was not to start it during the course of the trial. So whatever you have been doing before the trial, go ahead. Please wait for 2 months until this is over to start something else, because we really didn't want any confounding variables. We had an inclusion criteria, and the main one, we excluded kids with epilepsy. That was really the main. I think we had to exclude one child because of that. She was on anticonvulsant medications, and we decided to be careful. It's the first study. Even though this treatment is extremely safe, we wanted to be safe. So we had 2 main objectives. One is to demonstrate that optodynamic therapy, I really like this new name, or transcranial photobiomodulation, it's an effective treatment modality for improving symptoms of autism in children. And secondly, we wanted to validate EEG as a valid measurement of the effect of TPBM and a valid biomarker of autism. Let me just check. So before the treatment, before kids started receiving any treatment, we conducted childhood autism rating scales for placebo and active conditions, of course. And then after they finished 8-week trial, we did those rating scales again for placebo and active conditions. We collected 64-channel medical grade EEG, and we also conducted weekly parental interviews. So we called the parents and asked how your kids are doing. We also talked to the therapist once toward the end of the study, and that was also phenomenal. I'll talk about it later. So it was a randomized, double-blind, placebo-controlled study. On this picture, you see a little boy. His name is Anthony. He was one of the oldest participants. He was the most, if not the most, one of the most severe kids. He's completely nonverbal. He was brought to this trial by his father, who was crying, literally crying, because they couldn't handle it anymore. They were afraid that their landlord will kick them out of the apartment because the boy was jumping, and he was a large child. He was jumping up and down, screaming, yelling. Even when he was happy and not upset, he still had to jump to stimulate himself. And he was making really loud and disturbing sounds. And so the parents really didn't know what to do with this child. So he is wearing our device. You can see him. The device is very comfortable. We're very proud of it because one of the issues that everybody brought up is these kids are sensitive. How are you going to charm them into wearing this device? Turns out it wasn't a problem. It was so soft. The material that covers all the electronics is really soft and smooth, and children loved it. And actually, they kind of love being snuggled. So many of them even enjoyed it. So the device uses LED, so near-infrared light is being delivered through LED, and it stimulates very targeted brain areas. Specifically, we stimulate occipital lobe, we stimulate prefrontal cortex, we stimulate broken Wernicke, left angular gyrus, and some points on the default mode network. And we can talk about it later, maybe offline, why we believe it's important to stimulate those areas. But again, this is not a helmet that just shoots lights into your entire head. That would be the wrong thing to do. And after many discussions with our advisors, we figured out the specific points that need to be stimulated. It's infrared light, so the wavelength is 850 nanometers, and we used pulsing, 40 Hertz pulsing. So a little bit about our sample. 30 kids were recruited, were enrolled. 23 boys, 7 girls. We couldn't get equal number of participants, boys and girls, because this is just the nature of the disease. This condition affects boys more than it affects girls. And we really wanted to proceed with the trial rather than wait until we match them by gender. The average age was 4.5 years old. We did have older kids, 6 years old, and we did have younger kids, 2-year-olds. Complexion is important for this technology because darker skin just absorbs more light, and some of our darker participants weren't. At some point, I was worried that did we get all African American kids into the placebo condition? Nobody's responding. What's going on? By week 7 or 6, suddenly parents of kids with darker skin started saying, "Oh, I see a big difference." And this is because dark skin absorbs light, and when we think about dosage, this needs to be taken into account. We had 63% of our kids had some language. They again, these are moderate to severe kids. Some language doesn't mean functional language. They were echolalic. They couldn't express what they need, but they did have some language. And 37% were nonverbal. 43% received ABA. And 34% received something else, speech therapy, some kind of therapy, and 23% received nothing. This really broke my heart because these are moderate to severe kids in New York City, where therapy is really available, but still, for some people, it's more expensive. Some people didn't have time to bring the kid to the therapist. Some people didn't receive therapy through channels like Department of Education, they weren't approved. And so even for these severe kids, 23% of them were left with nothing, which is really, really sad. I just wanted to show, this is the demographics between groups. The main point here is that there is no statistically significant difference in the distribution between the groups. So we did have 5 girls in the placebo. Out of 7, 5 of them were in placebo condition and 2 inactive. With only 30 participants, this difference wasn't statistically significant. We were lucky with age. Active condition was 4.7 months and placebo condition 4.5 months. We were very lucky with therapy. It turns out 12 participants in active condition received some kind of therapy and 11 in placebo. So the groups were really well matched. I just want to point out that we had slightly more nonverbal kids. 7 nonverbal kids happened to be in active condition and 4 nonverbal kids happened to be in placebo condition. This difference also wasn't statistically significant, but it could be important. So the exciting slide, the results. Childhood autism rating scale. So this is, for those who don't know, this is the behavior. There are scales that measure eye contact, scales that measure response to name, response to commands, usage of language, usage of body, usage of toys, ability to play with toys, ability to interact with others. This is standardized scales that have been used in clinical practice for many years. This is well-validated method to diagnose autism and measure severity of autism. And that's why we selected it, because it's able to measure the severity of autism. And you can see statistically significant drop in the severity of symptoms for these children only in active condition. So if you look at the bottom chart on the bottom, you'll see that about 9-point drop was for active condition, and FDA considers 4-point drop to be clinically significant. So here we have more than twice as much. For placebo, we did have some placebo effect, about 2-point drop was placebo condition. So this is a huge change in behavioral symptoms. Oh, this is a fun slide. We talked to parents. Every week we called parents and we asked the same, not us, our research assistants who were blinded to the experimental condition. The parents were blinded to the experimental conditions. The blinding was done really well. So those assistants, we're forever thankful to them, called the parents and said, how is Anthony doing? How is Camila doing? They asked about eye contact, they asked about concentration, communication, any improvements in behavior. They also asked about side effects, headaches, tics, self-injurious behaviors. So first of all, every study, I'm convinced now, should have a placebo condition, because parents are extremely hopeful and parents really want to see improvement. And there is a lot of energy when you enroll your child in a trial. There is a lot of energy around them. You spend more time at the time, you interact more at the time, and that's great. However, to evaluate the effect of the treatment, we really need placebo control. And we did have significant difference even in parental interviews. We did have placebo effect clearly, but p-value was exactly at 0.05, which is statistically significant. And parents of participants in active condition reported those positive events more frequently. So this is simple counting, and it was non-parametric statistics. I believe it was Wilcoxon test. For side effects, there was no difference between conditions. So again, at least for the 8 weeks, for the duration of the trial, this is a very safe treatment. Nothing happened to the participants of active condition that was statistically or clinically different from the participants in placebo conditions. We did have a couple of kids who became more active or a little bit overactive, and that resolved spontaneously after a couple of days. Now moving on to EEG. So we looked at the intensity of delta wave. Remember the slide I showed in the beginning of the presentation that showed EEG data from Zamarodi? They found reduction of the intensity of slow waves because they used healthy adults. The slow waves was theta waves. Here we looked at delta waves, and I have to say that the very presence of delta waves in wakeful state is a sign of neurological condition. It shouldn't be there. These are deep sleep waves, unless the brain is inflamed, unless something is happening to the brain. I'll talk about it later, but for now, I just want you to look at the top chart on the top. And the line of best fit for the active condition is orange, and it shows negative slope. As these kids participate in the study, the intensity of the delta waves drops down. However, nothing like that happened to kids in placebo condition. Now, I want you to look at the chart on the bottom, and this is my favorite finding. I'm so excited. We correlated the change in delta waves with the change in CARS. So the y-axis shows you change in CARS, and 0 is the midpoint. For some kids got worse and their score went up. Some kids improved and their CARS dropped down. And the x-axis is the change in delta. So if you look at the bottom left corner, these are the kids who are doing the best. Their CARS is low and their delta waves is low. And you see majority of kids here are red, which is active condition. And if you look here, you see kids with high presence of delta waves and high CARS scores. This correlation was hugely significant. R squared is 0.64, and p-value is extremely small. What did we just do? We validated first of all, both our measures, right? We found intermeasure validity, which is hugely important. We also validated delta waves as a potentially biomarker of autism, because again, when there is something off with the brain and it correlates with the behavior, the intensity of delta wave is higher. When did this change in delta wave happen? Again, the presentation before me talked about also change in EEG and brain waves, and I think, if I'm not mistaken, they mentioned by week 2, by 14th day. We found the same here. By day 14, again, the red chart represents the intensity of delta, sorry, the red line represents the intensity of delta waves. We see statistically significant drop. Nothing happened, obviously, to placebo condition. And what's interesting is that the behavior changed by week 3 or 4. So when we analyzed the data, we noticed that change in delta waves preceded the change in behavior. We didn't know it as we were doing the study, of course, but after the analysis was complete, we noticed that first, there is electrophysiological change, at least in our trial, right, with 30 participants. I don't want to overstate the case, but it is what we found. So first, there is a drop in the intensity of delta waves by about day 14. And then in a couple of weeks after, parents become really excited and report changes in behavior. Now I want to talk about therapies, because also it coincides nicely with the presentation right before me. Therapists had no idea about this trial. We asked the parents, please, for the purpose of the study, do not tell the parents, the therapists, that your child is enrolled in a study. Just don't do it. And about week 7, close to the end of the participation in the study, obviously for active and for placebo conditions, we asked parents for the permission to contact the therapist. And we called the therapist and we asked, did you see any changes in Anthony or in Camilo or in Kira for the last 2 months? And the therapist said overwhelming response was the same: better eye contact and better focus, better responsiveness. Now these kids are no longer running around banging their head, for those who did, and they're not withdrawn, not looking at the therapist. They're there and they're willing to learn, they're able to focus, and they can interact. So I really think that this brain stimulation and brain modulation can reach its maximum potential in synergy with the therapy. Because for the therapeutic techniques to work, you need a child who is able to respond and receive. And this is what we want to give the therapist. I just want to mention that delta waves, we didn't come up with it. They have been identified as a possible biomarker of autism before us by other researchers. Again, delta waves are usually present during sleep, and they are called slow wave sleep. However, in wakeful state, they can be present if there is some neurological abnormality, specific microglial activation, inflammation, and they also are present in classical mitochondrial disease and autism spectrum disorder. For example, they have been found in Rett disorder, which is a type of autism spectrum disorder. So this is very consistent with the literature, and we found it in our randomized clinical trial, which is, I think it's great. We had a lot of happy parents. I could be reading these testimonies. I just want to say, instead of reading these testimonies, I just want to say that we had several kids who were relabeled by their therapists spontaneously. Again, the therapists didn't know the kids are participating in the trial. They relabeled the kids from severe to moderate and transferred them to the next group. So we had a 5-year-old girl who was severely autistic. By the end of the trial, her teachers in school convened and decided to transfer her to moderate group. We followed up with the mother. She continues 2 months later. She continues to study in the group for moderate kids, and she's doing really well. We had 2 identical twins, and one happened to be in a control condition and one happened to be in active condition. And that was actually heartbreaking for the parents who were bringing them both and who really wanted to help both of their children. But the child in the active condition progressed. The teachers were gushing about him because he started signaling that he wants to use the bathroom. He turned 6 during the trial, and so did his twin brother. He started communicating his needs. Bathroom wasn't the only need that he was communicating, but he started communicating through individual words, through signaling. He became more interactive, and that was a huge change in his life and life of his parents and in life of his teachers. Unfortunately, that didn't happen to his identical brother, who was in placebo condition. So where are we now? We just submitted for breakthrough medical device designation based on this data. We also just received yesterday a patent for the design of our device. And so we want to move on to phase 2 clinical trial. We want to test more kids. We want to expand the age of the kids up to 12 years old. Yes, we love neuroplasticity, but we believe we can expand this window. We want to prove that our device works best in synergy with ABA, and we want to do this comparison between kids who receive and do not receive ABA. We only used CARS for measurement. We want to use very specific measurement. We want to use at language outcome, at cognitive abilities, executive functions, and we want to look at various biomarkers. Most importantly, in our new version of the device, we want to collect EEG in real time with transcranial photobiomodulation to see exactly what's happening in the brain as light comes in. And we want to match behavioral outcomes with EEG data in much more specific and detailed level. And of course, the future is personalization of this device. For every child is different. Every child has her own or their own developmental trajectory, their own story, and we want to be able to make this available and effective for every child. I just want to say a huge thank you to our team. We have an amazing team of colleagues, scientific advisors who are leading us. And I want to say a huge thank you to Brain Foundation for inviting us to present. It's truly an amazing privilege to be a part of this group. This is just reference.
Thank you very much, Dr. Steingold. That was remarkable, and congratulations for the placebo-controlled study, which this field really needs to start getting that into the mainstream thinking, right? So there are several questions here, and I'm mindful of the time as well. One, if I could just start, is what is the patient allowed to do when wearing this device? Can they sleep, for example?
They could sleep, but they had to wear the device only for 5 to 6 minutes. So they didn't need to sleep. In fact, this is designed to be a mobile device. So the kids could move around, and that's what they did. They played, they moved around.
Okay, great. And then this one, I'm sure several people had this question, but are TMS and TPBM potentially synergistic, and would responders, should they respond to both? And would there be any advantage?
Fascinating question. The honest answer, data will tell. Maybe Dr. Fratkin could say more whether they could be used in synergy. There is not the honest answer. There is not enough data to tell you, precisely.
Okay. Sorry, go ahead, Dr. Fratkin.
Yeah, we know about different studies about TMS right now, but we don't have enough data on photobiomodulation yet. So, but we think that it will have similar results in the future, and we will go to at some point.
Great, thank you. And then the last question I have on the, oh, sorry, maybe it's not the last question. Are there any specific behavior issues that correlate more with delta wave abnormalities, such as scripting, attention problems, shimming, etc.?
So I think it's a fascinating question, and we have to mine data for more for this information. We have this data. We just finished the trials. We didn't look at that level of specificity yet, but I have to tell you, I think attention is because, again, there was something that all therapists reported to us, that kids became more focused. And I do think, and this is just my intuition, I have to do correlations, that the drop in delta wave and the increase in focus go together. That's just my answer today. I have to look at the data. If it changes, I'll let you know.
Sure, fair fair enough. It's early days, right? And the device is not available yet. Is that correct?
It's not available yet. It's been investigated. We're starting phase 2 clinical trial. As soon as we can, we'll start phase 2 clinical trial, and we can't wait to make it available. Actually, we see dire need. Yeah.
Okay. And so you mentioned the phase 2 or the phase 2 study design changes, and you're going to include sort of with and without ABA. Is that correct?
Yes, we are going to look at this condition separately because we believe it works best in synergy. So we need to prove it. So
you also mentioned at the very end that everybody has their own trajectory, which from a statistical point of view could equal high variability. And so does that trial become very large now in order for you to see a significant difference between ABA and no ABA?
We will have to speak to FDA about the sample size. But I was very surprised with the power of our 30 participant study and the effect size. So yes, there will be variability. I see that there is variability, especially because this is pediatric population. Whenever you work with a pediatric population that is developing brain, there is a lot of variability. But at the same time, I'm convinced now that this treatment is doing something quite powerful. So I think we will be able to overcome the variability.
Okay, fair enough. So I know we're kind of just about out of time here, and Pramila's online now again, or on video. So I should probably close it there. There's still a couple of outstanding questions, Dr. Steingold, if you wouldn't mind looking at them in the Q&A box and maybe onto those, that would be great. And again, thank you to you and to all the speakers in this session today. Remarkable diversity of efforts here, which is just really inspiring. So bright future, no pun intended.
Thank you.
Thank you so much once again, Stuart, for chairing the session. That was really exciting. And thank you so much to all of you panelists, participants, and speakers. And thank you to the attendees for sticking with us till the end. And as a mom, I'm really full of hope listening to all these talks over yesterday and today. We will be having a number of events next year as well. So please sign up to receive those notifications and also subscribe to our YouTube channel. We put out a lot of information there, and keep in touch, and we will continue this and see you all next year at Synchrony.
And thank you, Pramila. Excellent job again, as usual. Never a doubt there, but you probably deserve a good night's sleep after all this the last couple of days. So thank you.
Yes, thank you very much.
Thank you.
Thank you. See you.
Excellent. Well done.
Thank you.
Good job. Thank you, Pramila.
Thank you. Thank you on behalf of all of us here.
Thank you so much.